Welcome back, everyone. Now we're going to shift gears into mitochondria and the lung transplant, organ transplant, and you'll be hearing with me today for a panel discussion. We have Dr. Ori Rotstein, who is the Vice President of Research Innovation at the Unity Health Toronto. He is also a professor at the University of Toronto. Sorry, I lost my paper. And we also have here with us Dr. Anna Konvalinka, who was recruited in 2015 as a transplant nephrologist and a clinician scientist at the University of Toronto General Hospital at the Universe Health Network. And join us in a few minutes will also be Dr. Marcelo Cypel, who is a professor and a surgeon at the University of Toronto and Universe Health Network. I welcome you all for being here with me today. And now we are going to have a 5 minutes presentations from each of you, and let's get started from Anna.
Good afternoon, everyone. I'm really delighted to be here and to discuss this important topic. So my name is Anna Konvalinka, as you heard, and I'm a clinician scientist and transplant nephrologist focusing on solving clinical problems using molecular and systems biology. And some of the main problems that we're working on include the main reasons for premature kidney allograft loss, which are antibody-mediated rejection, fibrosis, and ischemia-reperfusion injury. We studied ischemia-reperfusion injury in the kidney sustained at the time of transplant, which is particularly relevant to the conference today. And while working with Dr. Marco Zeltzner, who is a transplant surgeon, we wanted to understand the mechanisms responsible for the beneficial effects of normothermic ex vivo kidney perfusion prior to transplantation. And Dr. Zeltzner pioneered a porcine autotransplantation model of normothermic ex vivo kidney perfusion to study its effects on kidney function and structure following transplantation. So we applied unbiased proteomics to understand how the proteome of porcine kidneys changes over time as a result of this intervention. In other words, as a result of normothermic ex vivo kidney perfusion compared to conventional static cold storage. And what we found was that normothermic ex vivo kidney perfusion resulted in preservation of mitochondrial proteins in pig kidneys compared to static cold storage. And this increase in mitochondrial proteins was associated with superior kidney function following autotransplantation. In contrast, static cold storage of kidneys after induction of warm ischemia resulted in decline of this group of proteins and inferior kidney function following autotransplantation. We have also recently examined kidney transplant biopsies from patients who had acute tubular necrosis as a result of ischemia-reperfusion injury and compared them to other causes of graft injury using laser capture and microdissection followed by unbiased proteomics. And we found that patients with acute tubular necrosis had increased glycolytic proteins, specific group of glycolytic proteins in the tubular interstitium, which particularly gets injured during ischemia-reperfusion injury, and increased different group of glycolytic proteins in glomeruli, further supporting a metabolic shift in kidney cells due to ischemia-reperfusion injury. Proximal tubular epithelial kidney cells are the most prominent injured cells in the kidney during ischemia-reperfusion injury. And these cells are full of mitochondria and are very metabolically active, second only to cardiac myocytes. So we have shown that proximal tubular cells alter their mitochondrial function in response to ischemic injury and become more glycolytic, even though they're typically used to fatty acid oxidation and using oxidative phosphorylation. We're currently studying how normothermic ex vivo kidney perfusion actually modifies mitochondrial protein expression and activity with the goal of translating these findings to therapies that can be delivered to the allograft at the time of transplant. And finally, ischemia-reperfusion injury is known to be enhanced in male compared to female humans and animals. And we determined that proximal tubular kidney cells have completely distinct metabolism and express different genes in males compared to female kidneys and cells, suggesting that this differential predilection to injury may be explained by the differences in mitochondrial nutrient utilization and function. And I will end with this, and I look forward to the discussion. Thank you.
Thank you very much, Anna. Ori, I pass on to you to introduce yourself and your work on mitochondria.
So first of all, thank you very much, Anna. Thanks for the invitation and for your team putting together the event today. Looks really exciting. So you did introduce me as a professor of surgery. I do have a little bit of a disclaimer though. I'm a surgeon scientist, but I'm not a transplant surgeon. So I may see like a little, a petunia and an onion patch perhaps, but in fact, no. So my major area of interest is in the area of trauma surgery and shock resuscitation. And Anna already introduced the concept of ischemia reperfusion. And when you think about shock resuscitation, that is a trauma patient who has massive injury and loses a lot of blood and then is resuscitated after that, you can think of it as a form of global ischemia reperfusion injury. And really, it's in that context that we've studied organ injury related to ischemia reperfusion. The target organ that we use in our research group is the liver and also the lung. Those are very relevant in trauma patients after they sustain hemorrhagic shock and are resuscitated. But that's the animal model that we've now translated that into. And, the one thing about ischemia reperfusion injury, Anna already talked about kidneys. I talked a little bit about liver, but, if you look at the pathophysiological process, it's relevant to a lot of important diseases, heart attacks, strokes, injury to limbs during trauma. So it's really a pervasive pathophysiological mechanism. And for that reason, it's obviously very important to study. So our interest comes in the area of how reactive oxygen species that are generated related to ischemia reperfusion injury might modulate the inflammatory response, and in particular become injurious to the tissues. And I think it's very well accepted now that mitochondrial dysfunction that occurs around the area of ischemia reperfusion actually is a major source of mitochondrial reactive oxygen species. And our focus has really been on how can we optimize the function of the mitochondrial pool in cells in order to actually optimize the quality of the mitochondria and therefore mitigate the effects of mitochondrial reactive oxygen species. And that's really the general thesis that we followed on. Now, interestingly, we came upon a very interesting intervention that had been used in a lot of circumstances called remote ischemic conditioning. And remote ischemic conditioning is when you actually put a tourniquet onto an animal or even in humans in advance of actually inducing a subsequent injury of ischemia reperfusion. And actually, remarkably, by that, the tourniquet being inflated and deflated on multiple circumstances and cycles can actually be protective in terms of the subsequent ischemia reperfusion injury. And our focus has really been in a murine model of hemorrhagic shock, where we showed that by performing remote ischemic conditioning on the hind limb of the animal, we can actually prevent the liver and lung injury that occurs related to hemorrhagic shock. And so that really was the context that we continued to do our research. And one of the questions we asked was, how might reactive oxygen species be modulated during the remote ischemic conditioning? And so we actually discovered that remote ischemic conditioning reduced the amount of reactive oxygen species, the mitochondrial reactive oxygen species in the target organ. And in addition to focus a little bit more closely on mitochondrial quality, we actually looked at the effect of remote ischemic conditioning on mitophagy, or the clearance of dysfunctional mitochondria. And that has really been one of the areas we focused on a little bit. My graduate student, Avinash Mukala, will be talking a little bit more detail. But in essence, what we have shown in our work is that remote ischemic conditioning, by a mechanism that he'll talk about a little bit, is shown to actually increase mitophagy in hepatocytes. And by increasing mitophagy, we actually clear dysfunctional mitochondria, improve the mitochondrial pool, and actually are protective in the organ dysfunction that occurs related to hemorrhagic shock. And also he extended that into a model of liver ischemia reperfusion, maybe a little bit more relevant to the particular transplant focus that we have today, but it is a pretty general type of mechanism that we're talking about. So fundamentally now what we're trying to do is better understand the regulation of mitophagy and the context of ischemia reperfusion injury, how we might modulate that, and by modulating that, how we can augment mitophagy in cells that are at risk from excessive oxidative stress from the mitochondria in the context of ischemia reperfusion. So I'm happy to take questions in the panel, but thanks again for the opportunity to give a few comments about our work.
Thank you very much, Ori. Now passing on to Marcelo. Thank you for joining us. I see you are in Boston live with us.
Thank you. Can you hear me okay?
Yes, we can hear you well. Thank you.
Yeah, sorry, there is a little bit of a music on the background here. I hope you are not seeing.
All good.
Hearing that. So I thank you for the invitation. And, I think, Ori has summarized a lot of the importance of the mitochondrial work in the, reperfusion injury scenario. And, I'm a clinical lung transplant surgeon. So lung transplantation success depends a lot on the, how the organs are preserved. And how rush our lives have to be as well depends on how long can we preserve organs. It used to be, that we, once the organ is removed from the donor, you have to really rush into putting the organ to the recipient, which has somewhat impacted the interest of professionals actually to do transplantation overall. And so we started looking at the mitochondria in the context of lung preservation over the past 2 years. And we have this great collaboration with Dr. Andreazza and the team. And one of the main discoveries we had was how the mitochondria injury is susceptible depending on the temperature that you preserve the organ. And the gold standard organ preservation has been to just put the organ on ice. That has been since the 60s when the first kidney transplant was performed, and then the first lungs in the 80s. And that's still the gold standard up to today. And it's very interesting that people didn't really reevaluated that concept over those many years. And so what we found is if you have a slight warmer temperature at 10 degrees, for example, you have a much better protection of the mitochondrial quality. You have less release of mitochondrial DNA. And as a consequence, you have less inflammation in the organ, which happens by mitochondrial DNA release, and activating the NLRP pathway, and then some of the cell death pathways as well. So that has been a huge mechanism that we found, and we did the large animal studies showing the major benefit on the functional level on these lungs. And we have moved on even to do a clinical trial to use these organs preserved much longer now. It used to be 5-6 hours. Now we are preserving lungs for 18 hours on the clinical side and transplanting them with very good success. So that's one of the major changes I would say over the past few years in the field. The other aspect that we're interested is the use of mitochondrial DNA as a biomarker of lung injury and correlation with ischemia reperfusion and graft dysfunction. And I have to say there is a lot of interest in this conference here in Boston, which a lot of the sessions are related, interestingly, not to mitochondrial cell-free DNA, but to cell-free DNA overall. Mm-hmm. In the blood of the patient after transplantation, correlation with episodes of rejection. Can we use this to differentiate rejection from infection? That sometimes on a clinical level is very difficult to differentiate. But we, again, based on the expertise, we're trying to focus more on whether the portion of the cell-free DNA in the circulation related to the mitochondria is the one that could be very important. So I think that's a summary, that I could give. And I think Erica is going to present the more details about that.
Mm-hmm. Yeah. Thank you. This is a great way to actually get this discussion started. So the hope now is that the audience join us asking questions via the Q&A button. So please send your questions away and we will come discussions. But to start kicking off the discussion, I can see from each of you the relevance of the mitochondria has played of either improving the conditions that you are treating on your lab or preserving better the lungs or increasing the importance of reperfusion therapy. So I'm going to start with a very open-ended question. How do you see the future of research in terms of mitochondrial research in each of your areas? Do you see that as more a secondary area of research, or are it going to become an essential area of understanding to either prevent or to treat the patients which you see in your clinical days? Maybe I will start with Anna.
Sure. Thank you for that very relevant question, Anna. So I can start with how we are specifically going to be addressing the issue of mitochondria and using that to fuel further development of therapies for organ transplantation, but also how I see mitochondria playing a role globally in transplantation. So as is, I think, probably well recognized to the participants of the conference, mitochondria play a critical role in ischemia or perfusion injury that takes place at the time of any transplant. Mm-hmm. And that's really due to kind of briefly accumulation of succinate in mitochondria and the reversal of complex 1 in mitochondrial membrane pumping electrons the wrong way and resulting in rapid and massive superoxide generation. And this superoxide generation basically leads to a cascade of downstream negative effects. And so our interest really started because although Marcelo is really a pioneer in normothermic ex vivo organ perfusion, so I've worked with Mark Zeltzner, who has been looking at kidney normothermic ex vivo perfusion, and we see similarly benefits in terms of organ function and structure following transplantation after this method of storage compared to the gold standard static cold storage. And we've really discovered that these groups of large groups of mitochondrial proteins, mitochondrial matrix and the intracellular mitochondrial proteins are really maintained in the course of normothermic ex vivo perfusion and following transplantation, which is not the case when the kidney is stored on ice. And we have come to realize that the most likely sort of transcription factors or transcriptional regulators of these are this PGC1 alpha, mitochondrial biogenesis regulator, but also some of the PPAR transcription factors. And one other very interesting nuclear factor that I won't talk about too much now, but that we are now interested in targeting. In other words, developing agonists for these particular transcription factors that can be delivered at the time of or prior to organ transplantation in order to improve mitochondrial function. So that's how we are mainly looking at this. in general, the field is huge and this area has really grown. There are various agents that are targeting this succinate accumulation within the mitochondria, this MitoQ molecule that also binds all reactive oxygen species. And other people who are approaching it similarly like us with trying to target these major mitochondrial biogenesis factors. But also in transplantation, mitochondria play a much bigger role, and some of these briefly include immunometabolism, so T cell metabolism, where maintenance of oxidative phosphorylation is really critical for maintenance of T regulatory cells relative to T effector cells, which upregulate glycolysis massively. And importantly, in the kidney, and I'm not sure if that's the case in other organs, but in the kidney, actually maintenance of fatty acid oxidation and oxidative phosphorylation actually is critical to preventing fibrosis. So that some groups have shown that even just agents that either genetic or pharmacological manipulation that results in maintenance of fatty acid oxidation within the kidney prevents fibrosis. So I think there are many avenues, and clearly mitochondria are very important, and I think there's lots of now interest and focus on this.
Fascinating how to see how just a slight change on temperature is actually changing the mitochondria from an inert to actually a more metabolic active, which potentially is producing less damage. Or do you want to comment about a little bit on your perspectives as well in terms of how mitochondria will progress in your area of research?
Yeah, I think, thanks, Anna. You asked a very open-ended question, and it was about the future. So I guess I'll agree with Dr. Konvalinka. I think the future is very bright. I think people are now over the idea that the only thing the mitochondrion does is maintain ATP. I think people know that it's broadly impactful on virtually all aspects of cell function. And being able to modulate it can have tremendous impact on the cell. I think the other point that's important is it's not just transplant. Ischemia reperfusion injury is a common pathological process that's pervasive across medicine. And so being able to address that disease by modulating mitochondrial function in some aspect or another can have incredible impact. And I think by understanding the process of mitochondrial quality control, whether it's to get rid of dysfunctional mitochondria or to generate new functional mitochondria, to be able to understand that and to be able to do it in the in vivo setting has potential therapeutic impact and very important ones. And I think that that's really the focus of our work. We're studying biogenesis as well as mitophagy, but when you think of developing molecules like Anna said, that might be able to induce biogenesis or to increase mitophagy and clearance of dysfunctional mitochondria, those are things that would be obvious important potential therapeutics for a number of disease processes very broadly across medicine.
Oh, fantastic. Thank you. Marcelo, what is your thoughts?
Yes, I think, again, I think Anna and Ori covered a lot of the potentials for the future, which overlap with the potentials in the field of transplantation as well. So I think, again, going from, options of using as a biomarker, I think that's an important one. I think as we discussed before, the haplotype matching is something that hasn't been explored yet in the field of transplantation, could be something interesting. I think the mitochondrial transplantation has, I think, makes all sense for organ preservation. I mean, it's really, organ preservation injury is related to, lack of ATP and some other features that mitochondria is protecting in the functioning of the cell. And maybe finally, we can also, one of the questions is, as we have these organ preservation systems as well, one thing, of course, is to try to protect the mitochondria at that point, but maybe we could also try to counteract some of the injury that is happening. So I always, also wondering if we have this mitochondrial cell free DNA released in the liquid of perfusion, is this only a biomarker, or this is amplifying the injury in the organ during preservation, right? And maybe there are ways to protect the endothelium and, other therapies directed to the organ that is not a direct effect in the mitochondria, but it could also help on that setting. So I think there is a lot of, lots of studies, and it's quite promising, I would say.
Yeah, it's wonderful to hear you all speaking, because it kind of highlight one of the main missions of MitoGI, which is really enhancing and understanding better how mitochondria plays a role in health, but also in disease, and through that develop better diagnostic and targets. So I want to spend the next few minutes discussing with you the relevance of mitochondrial diagnostics. So let's say, or maybe I started this with you, because you mentioned something about the reperfusion and the damage that I think it is fantastic for the views of mitochondria. Let's say in the future we'll be able to have really great mitochondrial diagnostics. Do you think this would help also identify these patients that are at risk and potentially prevent, or maybe even by diagnosing, ameliorating faster or with a more targeted treatments for them? What are your thoughts on mitochondrial diagnostics and preventions for reperfusion and ischemia?
Well, since none of us is answering, I'll go first.
Okay.
So I guess as a broad answer, the promise of personalized medicine is that we can define something we can measure that would allow us to better target therapy. So if we're able to potentially define some, mitochondrial diagnostic, let's say mitochondrial DNA in the blood or mito-ROS, that may allow us to actually target the specific type of intervention that we want to do. Do we want to, increase clearance of dysfunctional mitochondria? Do we want to clear mitochondrial DNA from the blood, et cetera? So I think that there's, very much, a diagnostic potential for measuring some aspects of, mitochondrial function. Certainly I don't have expertise on that, but those are just two that I think we could point to right away.
Yeah, fantastic. And when you're speaking about the target treatments, I was very interested in you speaking about the PGC1 alpha and the PPAR gammas. I think those are extremely important pathways of controlling mitochondrial biogenesis. There's so many studies have been done with bezafibrates and other fibrates that control this. Have you guys used those in the settings of your studies, or any other molecules targeting the pathways?
Yeah, that's interesting. I'm not allowed to. I'm not sure if I'm allowed to say, but I'm currently developing a proposal with one of the medicinal biochemists at U of T to use one of the agonists of PPAR alpha, one of the newer and sort of safe agonists of PPAR alpha that's not been used previously in this context. And we'll try first on Dr. Zeltzner's normothermic ex vivo kidney perfusion to deliver it. And we'll also try to see if it works, even if just given when the kidney is on ice, which I'm less optimistic about because there's simply less mitochondrial function or no mitochondrial function. So that's basically how we are going to try that next. And there is this new also nuclear factor called HNF4 alpha, hepatocyte nuclear factor alpha, which is 4 alpha, which is really important in kidney development, and which we think may be fundamentally involved in this kind of favorable response of mitochondria. So we're looking for ways and for novel agonists to also use in a similar fashion to test the time of transplantation. So that's basically how we have approached this field. Fenofibrates certainly have been tried and they are used clinically. They're not the most favorite drug because of some of their renal negative side effects for kidney ischemia or perfusion injury. But certainly in these big important papers that have shown a preservation of fatty acid oxidation and thus prevention of fibrosis. Fenofibrates have been used successfully in that context. So I think, we need novel therapies along those lines. I personally believe very much in a systems approaches. So, from a diagnostic perspective, we are proposing to look at the same time when at the time of transplant we all get to do a kidney biopsy on the kidney allograft. So we're proposing to measure mitochondrial function in real time using seahorse, which we can do now, to look at protein expression and then at the same time combine that with measures of metabolites in the blood. And so sort of to use some kind of machine learning algorithm that can, potentially put that in the context in the future. We think there is a promise in looking at metabolites in blood. I mean, even these studies that discovered succinate, for example, they, people were looking just at unbiased metabolites that are increased in ischemia or perfusion injury across different organs. And that's how succinate was really discovered first. So I think there is promise in that.
Thank you. Marcelo, you talked very briefly on your opening about mitochondrial haplogroups, and I know our group Amy studied that a little bit. Can you comment a little bit more? What do you think this potential of the haplogroups in terms of mismatching or matching in terms of organ transplant, in your case in lung transplant?
Yeah, I mean, I think that's just an idea right now. It's something. It's a very new concept. So, there is very preliminary work, I believe, in kidney that had shown some type of haplotype to be a higher risk of PGF, but no one has looked at the combination of different haplotypes. Interestingly, there are some. Again, preliminary data in heart transplantation that is showing, and again, I just saw this in the last couple of days, that some of the increased risks that we see when we have a donor of one specific race transplanted to, donor recipients of different races, is that an increased risk of reperfusion injury in that setting, or more alloimmunity after transplantation, increased alloimmunity may be related to the differences in mitochondrial DNA characteristics, and that's probably related to the genetic background, so, again, this is very preliminary. We still don't know. And just again, to follow up on your initial question about the diagnostics part, I think a very good and interesting part is especially the cell-free DNA is very dynamic. So we are seeing that, you have that when you have, an acute event, for example, post-transplant, like people with COVID ARDS had like huge amounts of, just to put in context, of cell-free DNA in circulation, like 100 times higher than we see in rejection for transplantation. Just so to have a bit, but it goes down very quickly as the process resolves. So I think it could be an interesting aspect, again, to look at not only one time point, but the dynamics of the marker. Yeah.
That's fantastic. Just to put in for our audience, mitochondrial haplogroups are the parts of the mitochondrial DNA that is preserved through our anthropology. So it came from your ancestry, passed from your mom to your mom to your mom, and goes down until you get to you. And so we can trace where we come from. Depending on the type of a haplogroup you have, you might have a mitochondria that is more efficient, or a mitochondria that is a little bit less efficient in energy production. So I think the concept, which is exactly what Marcelo saying, is very big in the hypothesis, there is no data yet, is if you actually transplant a mitochondria that is high efficient into an organ that has a mitochondria that is low efficient, would that work, or would that be a problem? It's like having a car that runs in diesel and you put in gasoline. How this is going to happen, right? So I think this is a future of promising in the field, and maybe not only for lung, but kidneys, hearts, and liver. I think that's a story to still to be told. On the last 15 minutes that we have, I'd like us to think again on the future. And I want to go through each of you for final comments and ask, what do you think, in your view, would really help your group to excel the research you're doing? Is collaboration, is funding? What is necessary right now for you that you think that we could help you to achieve that? I'll let anyone start, but if not, I'll call the name.
I'm happy to start.
Great, thank you.
Yeah, I think. I think the collaboration is huge, and I think we're very well posed also in Toronto to have, several groups of different expertise, and of course, funding is very important as well. I'm very optimistic about the field of the mitochondrial transplantation area. I think this will likely become powerful in the transplant setting, which is the area I work again. So I won't comment too much about other potential applications that the team here is working. But I think having to develop very reproducible protocols, clinical grade, understand dosage. All those things I think are the main goals for the next 5 years, I would say, that we can use this as a therapeutic arm.
It's fantastic. Thank you, Marcelo. Ori, would you like to go next?
Sure. So I think the most important thing is having an event like this.
Awesome.
Really, the networking and collaboration from the Mito2I has been actually huge for our group. And already, I'm going to call Anna tomorrow, and I'm going to find out how I can get one of those drugs to use in our ischemic reperfusion model. I'm going to go and I'm going to learn more about mitochondrial haplotypes so that I can, ask my student Avinash to kind of give the very best type of mitochondrion in his mitochondrial transplant study. So I think that this is really a great start for us to get together and kind of share ideas and, share ideas with our students and your students and even get some more exciting projects out of it. So that's what I see as one of the really important results of this, but also an important go forward.
And you couldn't call me on a better time because the drugs are just arriving tomorrow in the lab. So we're happy to share. And the same goes for you if you want to test some of our drugs, it would be lovely. And your final thoughts for us?
Yeah, no, I agree with everything that's been said. I think both collaboration and funding are critically important. My team really enjoys collaborating with lots of diverse different teams. And I think I agree with Ori that this type of venue is perfect in kind of getting ideas started and, getting collaborations. So clearly we need experts in mitochondrial biology, but also, I think collaborations with medicinal biochemists and even pharma companies could be potentially very helpful. Of course, nothing can be done without funding, and certainly these organ reperfusion studies, for example, both in pig models and in humans, are extremely expensive.
Mm-hmm.
And so, so I think funding, we definitely need funding to accelerate research.
Thank you. I'd like to thank you all very much for being here today. And I really want to expand the thanks to St. Mike's to have supported us with the graduate scholarships. And Marcelo, in your behalf, I will also thank UHN to support our funding scholarships. It's been fantastic to see this progress coming, and hopefully we have more events like that over the year, perhaps a series of workshops, an idea that come to me that might be beneficial for all of us to continue this discussion. Thank you again very much for being here.
Thank you.
Thank you.
And thank you. Thank you for organizing.
Yeah, thank you for inviting us.